Hi, welcome to the MedLit Hour. This is my very first episode. My name is Ryan. I'm a first-year medical student, and I will be taking you through the clinical features and diagnosis of human monkeypox. Thank you so much for being here. Please give this video a thumbs up, like, and subscribe. The premise of this podcast is to go over very cool medical cases in published literature. I figured that the first episode should be something that is relevant currently in our world today. So I hope you guys enjoyed this presentation, and please give me feedback if you can, and thank you so much. A few disclaimers: this is not medical advice. First and foremost, if you are showing signs and symptoms of the monkeypox virus, visit your local emergency room. Number 2, all information provided is published in literature or provided by the CDC. Number 3, all information may be subject to change as more research is conducted. Number 4, slides may contain sensitive photos which some people may find disturbing or offensive. 5, always refer to state and federal agencies along with the CDC and the WHO for more information regarding about monkeypox. Lastly, monkeypox is not only transmitted among individuals who are part of the LGBTQIA+ community. In this episode, we are going to be looking at the following sections. We are going to go through an introduction, then followed by what's an orthopox virus, breakout at a glance, where we are currently, transmission, signs and symptoms, diagnosis, TNF deletions, vaccinations, treatments, chemical structure of the virus, morphology, pathophysiology, cell-mediated immune response, as well as long-term effects. Okay, so you might be thinking, what is the monkeypox virus? Well, it is a zoonotic disease belonging to the orthopox virus genus. That is how it is classified. It is clinically and structurally like variola, which is smallpox. While it was first recognized in monkeys in 1958, given the name, many rodents and squirrels have also been responsible for human monkeypox virus outbreaks. First case of human monkeypox was detected in 1970 in the Democratic Republic of Congo, called Zaire at the time. At this time, smallpox was still endemic, so many people were vaccinated against smallpox. This vaccine is estimated to give about 85% protection against the human monkeypox and prevented deaths. Human monkeypox mainly affected children younger than 15 who were in contact with an infected animal. And in 2003, infected exotic pets were imported from Ghana into the United States, causing the first human monkeypox outbreak. These exotic pets were placed with prairie dogs to be sold as pets and resulted in 53 cases in the Midwest. Further testing revealed that a Gambian rat, rope squirrel, and dormouse from the shipment were all tested positive for human monkeypox via PCR. What's an orthopox? Orthopox is a group of large double-stranded DNA that replicate in the cytoplasm of their own host cell. These viruses can include variola, which is smallpox, cowpox, and vaccinia. Vaccinia is the live virus in smallpox vaccine and provides a degree of protection against other pox viruses. Orthopox viruses are keen to evolutionary changes and adaptations, since some genes in their genomes will be intact in a certain virus but fragmented or deleted in another. This character in their genome gives them the ability to potentially reemerge as new virus species. Furthermore, it is observed that pox viruses undergo duplication and amplification in parts of their genome when faced with survival pressures such as antiviral measures. Later on in this episode, we are going to talk about TNF deletions and how this is relevant. Certain genes in their genomes can quickly mutate, giving them the ability to adapt to new environments, including new hosts. Cowpox has the largest genome and therefore contains all the genes found in the other viruses, whereas variola, which is smallpox, has the smallest genome. This just kind of shows the likelihood of a new variant with longer genomes to basically emerge as evolution goes on. I have been following monkeypox essentially since the beginning of this current breakout, which happened in May of this year, and I actually found some pretty alarming data. This is directly from the CDC, and I figured that I would put it in a chart to view better. So on the left, you have a bar chart of confirmed cases in non-endemic countries. This includes UK, Spain, Germany, and the United States. And what they found is that within a 2-week span, May 13th to June 2nd, they were able to confirm 780 cases in non-endemic countries. And if you look at the bar chart to the right, you will see that these are the cases that are in endemic countries, such as the Democratic Republic of Congo, Nigeria, Cameroon. And within a 6-month period, January 1st to June 1st, they were only able to confirm 44 cases, suspected 1,400 cases, and have had 66 deaths. I find these numbers highly interesting because it really gets you thinking about resources, what's available, treatments, vaccines, and just testing overall as to why they are not able to confirm as many cases as the United States, UK, or other countries that are non-endemic. I believe these numbers are very eye-opening as to kind of just showing the resource availability in various parts of the world. As of November 4th, we have 78,229 global cases. 77,301 of those cases are in non-endemic locations, and the United States currently has 28,657 cases, over 5,000 of them being from the state of California, 2,700 of them being from the state of Texas, another 2,700 being in the state of Florida, and 4,100 cases being from New York. On the following map, all of the states that are colored in dark blue indicate that there are more than 500 cases. Transmission can occur from an infected animal or human via saliva, respiratory droplets, skin contact, and bodily fluids. Human-to-human transmission of monkeypox is less contagious than smallpox, but previous research shows that it can occur in up to 11.7% of households without prior smallpox vaccine. Pregnant women can also transmit monkeypox via the placenta. In the past, the most amount of sequential human-to-human transmission cases have been up to 6 individuals. In endemic areas, especially in earlier outbreaks, men were at a higher risk of being infected with monkeypox due to the higher incidence exposure to infected animals. Later in this episode, we will talk about vaccinations. Individuals who receive the ACAM 2000 vaccine will be contagious and potentially be able to transmit the monkeypox virus to those who are unvaccinated. Patients with monkeypox present with fever, rash, headache, and fatigue, and a differentiating symptom from smallpox is lymphadenopathy, which are swollen lymph nodes. In the first couple of days, mucosal lesions develop, followed by skin lesions on the face, extremities, and may spread to the rest of the body. Over the following weeks, the lesions change in appearance around every other day and are described as different phases. First, flat and non-palpable macular lesions appear. These then raise into solid pink and discolored papular lesions. They are then followed by the trapping of fluid, becoming vesicular lesions, and finally followed by pustular lesions, which closely resemble pimples. These pustular lesions usually last around a week before crusts begin to form over the next following weeks. Once the clusters fall off, the patient is no longer considered infected. In most cases, patients usually recover 3 to 4 weeks after the initial symptom onset. Monkeypox cases can look different on everyone because the number of lesions can vary. Patients may present with these symptoms in a varying order. Some may present with lesions without fever, and some may present with different types of lesions. While mucosal lesions remain the most common symptom, some patients may present with lesions in other parts of the body, including genitalia or anal rashes. I have provided 3 images of patients who were diagnosed with smallpox as well as monkeypox, showing that the main differentiating symptom is going to be the lymphadenopathy. You will not see swollen lymph nodes in smallpox. However, you will see swollen lymph nodes in monkeypox cases. Research suggests that this may be indicative of a stronger humoral response to the virus. If you suspect that you have a case of monkeypox, history taking is very important in this case to understand the potential risk and exposure. Understanding of recent travels, leisure activities, and or providing care to someone who might be infected, along with clinical findings, play an important role in diagnosing monkeypox. Viral DNA in lesion crusts can be obtained for polymerase chain reaction to test for the presence of the monkeypox virus. As you can see from this table, the PCR is going to be the most effective and accurate way to diagnose monkeypox at the time. Other more advanced techniques like electron microscopy and immunohistochemistry are available in well-sophisticated labs. Anti-orthopox virus immunological assays may be used to detect antibodies to determine a previous exposure to a wide variety of orthopox viruses, but it will not provide a definitive diagnosis. Otherwise, serological assays that measure anti-orthopox virus immunoglobulin M, IgM, are more effective in diagnosis of these infections and will also be able to detect previous vaccinations. Tetracore orthopox biothreat is an assay that has accurately detected vaccinia and monkeypox. Again, vaccinia is the virus strain that is used in vaccinations against the smallpox virus. One of the most interesting findings is this TNF deletion. And basically, TNF stands for tumor necrosis factor. It is a type of pro-inflammatory transmembrane cytokine. They are found at the cell surface and released as cytokines using a TNF converting enzyme. They are released by different types of cells like macrophages, natural killer, and T cells, as well as mast cells, and are important in mediating certain communications between the cells. TNF is responsible for a wide variety of tasks, including activating inflammatory genes, cell differentiation, apoptosis, as well as playing an important role in mediation of acute inflammation during viral infections. After a virus enters a host, TNF is induced locally. It will then enhance its own expression along with other pro-inflammatory cytokines and will secrete chemokines to provoke the recruitment and activation of leukocytes. This kind of response from TNF is crucial for limiting the spread of the pox viruses. However, pox viruses have implemented various molecular mechanisms to counteract this TNF antiviral activity. One distinctive mechanism of immune evasion is that they will secrete viral proteins from infected cells and bind it to the key host cytokines before they have the chance to even engage with cellular receptors, making them as decoy receptors by essentially binding and neutralizing TNF cytokines. To be more specific, pox viruses will encode two types of secreted proteins that will target the diverse members of the TNF ligand superfamily. They encode viral TNF receptors, which are functionally and structurally homologous to cell receptors, and they will also secrete and encode novel viral TNF binding proteins. The TNF gene is the target for the West African monkeypox and the monkeypox real-time PCR tests. This means that patients who have a TNF deletion will not be able to test via the real-time PCR because the TNF gene receptor is gone. If you have a highly suspicious case of monkeypox that was tested negatively, maybe consider that your patient might have a TNF deletion and refer that sample to the CDC so they can test it using an assay that is not based on the variola orthopox. I do want to say that these cases are very rare. The CDC is aware that there are 3 cases in the state of California in patients who have this TNF deletion. The schematic diagram here in this slide shows TNF cellular response and viral counteraction. TNF ligand superfamily has 19 structurally similar ligands that bind to type 1 membrane glycoproteins at the cell surface. This occurs at the TNF receptor and triggers the signaling pathway for this protein. TNF receptor 1 and TNF receptor 2 belong to 2 different subgroups of the TNF superfamily due to the differences in their intracellular signaling domains. The TNF receptor 1 cytoplasmic domain anchors with it a death domain denoted as DD on the schematic diagram. This enables direct interaction with the TNF receptor associated death domain denoted as capital TRADD in response to TNF binding. This complex of TNF receptor 1 along with the TRAD serves as a docking platform to recruit different types of signaling proteins such as the receptor interacting serine/threonine protein kinase 1, which is denoted as RIPK1, which is a kinase, so it's going to add phosphate groups. This is going to become phosphorylated, and it will initiate apoptotic cell death via the caspase cascade. On the other hand, you have TNF receptor 2, which promotes cell survival and does not contain a death domain. This will bind to the TNF receptor associated protein TRAF and activates nuclear factor Kb, denoted as NFKB, which is a transcription factor. TNF receptor 1 can also act as a pro-survival receptor when it is using the TRAD/TRAF2 along with the RIPK1 complex, which means in summary, TNF pro-survival action can be executed either through the TNF receptor 1 or 2, while TNF-mediated cell death only occurs through the TNF-R1 signaling. There are currently 2 vaccines available in the United States. The first one is called Jynneos. It is a live but attenuated vaccine, meaning that the live strain was weakened in a lab to limit or completely shut down replication. Jynneos is non-replicating and it is given in 2 separate shots, each one containing half an ml. It is made by a biotech company in Denmark and produced from this strain modified Vaccinia. Vaccinia has been traditionally used over the years and has been modified ever since then. So this is just another modified version of the vaccine we have used in the past. Interestingly enough, this strain was grown in primary chicken embryo fibroblast cells, which is commonly used in virology. After an individual receives both doses, it is designed to provoke a humoral and cellular immune response to the orthopox viruses. Currently, there is no data for patients under 18, pregnant women, or long-term effects. And while it is FDA approved and has significant data to prove that the vaccine does provide protection against the virus, there is no data to know how long the patients are protected. The second vaccine is more intense and it is called the ACAM2000. This is a live replicating vaccine that is administered vigorously than the other standard vaccines. It is given by using a 2-pronged bifurcated needle. The needle is dipped into the vaccine and then pricked into the skin several times until there is a presence of blood from the arm. This virus will grow at the injection site, causing a localized infection known as the take. After 2 to 5 days, a red, itchy lesion will develop and eventually turn into a blister. This blister will become a scab, fall off, and leave a scar. This takes place over the course of 3 weeks, and the response is evidence gaining protective immunity. This is seen in patients who have not been previously vaccinated. First-time vaccinees who don't display this response indicates a non-take and is deemed as a vaccination failure due to either impotent vaccine or inadequate vaccination technique. However, any prior vaccination may modify or reduce this response with revaccination. This means that the absence of a cutaneous response does not necessarily indicate a vaccination failure in those who have already previously been vaccinated. And these patients who do not have a cutaneous response upon revaccination do not require revaccination to try to elicit a cutaneous response. ACAM2000 is a live vaccinia virus. Individuals who receive this dose can actually transmit the virus. Actual deaths have been reported in unvaccinated contacts accidentally infected by individuals who have been vaccinated. Serious complications may follow either primary live vaccinia vaccination or revaccination, including myocarditis, pericarditis, dilated cardiomyopathy, encephalopathy, severe vaccinal skin infection, blindness, and fetal death in pregnant women. People who have known cardiac diseases have a higher risk of adverse effects. Additionally, patients with 3 or more of the following conditions have increased risk as well. This includes high blood pressure, hypercholesterolemia, diabetes, history of smoking, and other cardiac related history. As far as treatment goes, there are currently 2 medications that are FDA approved to treat smallpox. Even though they are not FDA approved to treat monkeypox, it has been shown to have some kind of efficacy when it comes to treating that particular virus. The first one is brincidofovir. It is a nucleotide analog that inhibits viral DNA polymerase. It was developed specifically to treat double-stranded DNA virus infections. It may also treat retinitis in patients with AIDS. The second medication is called tecovirimat. It is also FDA approved for smallpox as mentioned previously, and it inhibits intracellular release of the virus. For the chemical structure, a complete virus particle that consists of an RNA or DNA core with a protein coat called a capsid is called a virion. It is the extracellular infectious form of a virus, essentially when it has not yet hosted a cell. They predominantly contain structural proteins and enzymes with complete transcription capabilities via RNA polymerase. They can also undergo replication and release proteins in their genome. They rely on a host ribosome for mRNA translation, and the genome of each particle can be found inside the nucleosome within the core, surrounded by a lipoprotein envelope. Each particle can carry about 100 out of the 200 potential virus-encoded proteins. The virions vary from 220 to 450 nanometers in length and 140 to 260 nanometers in width and depth. They are often brick-shaped and appear as smooth, rounded rectangles. The core is dumbbell-shaped and surrounded by a sequence of membranes of about 30 nanometers thick. On a negative stain, a mix of virions will appear with short surface of 10 nanometers tubules referred to as mulberry forms, denoted as M. But a smaller number of particles are larger and more electron dense with a thick 20 to 25 nanometer membrane referred to as a capsule form, which is denoted as C. Morphogenesis begins in the cytoplasm during viral replication. Shortly after infection, the early cells have a crescent-like membrane structure that develops into circular structures via primary transcription, referred to as immature virions. The bilayer surface membrane on these virions will differentiate, forming an outer membrane and a core membrane. Together, this forms an intracellular enveloped mature virions, called IEVs. This mature virion then moves along the cellular microtubules to the cell surface where actin polymerizes behind it. They then exit the cell through specific microvilli by fusing the outermost lipoprotein layer with the plasma membrane, resulting in the release of mature virion within the inner lipoprotein layer, and now becomes the extracellular enveloped virion called EEV. Both mature virions and enveloped virions are infectious particles, each with specific surface antigen properties. However, both enveloped and non-enveloped forms achieve the same result. First, the mature virions will be fused onto the plasma membrane, causing the entry and uncoating of the particle. This releases viral content into the cell, initiating transcription of early, intermediate, and late class proteins. The production of these viral proteins regulates the host immune response and results in the subsequent assembly, morphogenesis, and release of future virion particles. Poxvirus infections evolve either as localized, fairly benign skin infection or as a systemic infection. Systemic infection results in viral dissemination with other formation of generalized skin lesions and usually involves some degree of morbidity and/or mortality. The general model for systemic orthopox pathogenesis is that the virus enters the host through a route that can be respiratory, mucosal surface, or break in the skin, and it replicates locally. The virus then spreads through the local lymphatics, causing a primary viremia, which is the spread into the blood. It subsequently spreads into the reticuloendothelial system, a system that contains monocytes to perform phagocytosis, which is mostly found in the liver. Viral replication in these organs results in secondary viremia. Symptom onset correlates with secondary viremia, leading to 1 to 2 days of symptoms such as the fever and lymphadenopathy before the lesions start to appear. Infected patients may be contagious at this time. Lesions start in the oropharynx, then appear on the skin. Serum antibodies are often detectable by this time. Due to the appearance of the lymphadenopathy, it is theorized that the host immune response involves all arms of the immune system. Complement, interferon, natural killer cells, and inflammatory cells are implicated in the early innate response. Pox-specific antibodies, including neutralizing antibodies, are subsequent components of the humoral response, and pox-specific cytotoxic lymphocytes are involved in cellular clearance of the infection. To retouch on what was previously said, various poxvirus-encoded proteins will bind and interfere with the function of the host cytokines, chemokines, interferon, as well as other cells. This may halt the apoptosis and prolong the life of the virus. An effective cell-mediated immune response requires natural killer cells, cytotoxic T lymphocytes, and many more to rapidly identify and eliminate infected cells before the virus can replicate and spread. Unfortunately, viruses have developed new mechanisms to reduce the efficiency of the CMI response. One mechanism seen by poxviruses is the downregulation of cell surface receptors. As previously discussed, we know that poxviruses can inhibit pro-inflammatory cytokines such as TNF. TNF also plays a role in regulating MHC expression. MHC stands for Major Histocompatibility Complex and is a wide range of cell surface receptors that help with the immune system recognize self from non-self by presenting foreign antigens to the CD8+ cytotoxic T lymphocytes. A decreased expression of class I MHC molecules has been observed following a poxvirus infection. Poxviruses alter the expression of other cell surface molecules that play roles in immune regulation. For example, downregulation of CD4 expression has been demonstrated following myxoma virus infection, which is also a poxvirus. If infected cells are recognized and targeted for clearance, poxvirus utilize a wide range of mechanisms to disrupt both extracellular cytokine networks and intracellular signaling cascades that transduce death signals associated with a functional cell-mediated immune response. Severe consequences were found to be more common among unvaccinated than vaccinated patients. Pitted scarring is the most common long-term complication of the infection. However, more serious complications can arise. A second common complication includes ocular infections, which may result in corneal scarring and permanent vision loss. This was also seen in patients who received the live strain ACAM 2000 vaccine. Patients have also been observed with pulmonary distress or bronchopneumonia, often late in the course of illness, suggesting a secondary infection of the lungs. That concludes today's episode. Thank you guys so much for listening. Here are all of the citations that were used to put this presentation together. Thank you again so much. Please subscribe and thumbs up if you are watching this on my YouTube channel. Thank you so much.